Hi, I'm Sricha Kamal, and this is my presentation on investigating the gene SPR2 and reproductive decline, and this is an epigenetic inquiry into germline gene regulation inside of C. elegans. So, just as some background on epigenetics, I'm sure you all know that inside of all of our cells we have DNA. And the way our DNA compacts itself is it wraps around a spool-like structure called a histone. Now, when it wraps around this histone, it can either loosen up, allowing for more active gene expression, or it can tighten up, not really allowing for much gene expression. And whether our genes are active or inactive is often determined by external epigenetic factors. And these are just factors that don't alter your underlying DNA sequence, but do determine which genes are on and off, and determine how genes are expressed, basically. So some examples of epigenetic regulation are DNA methylation, which is the additive of methyl groups onto our histones, which can cause chromatin and DNA to kind of tighten up, not really allowing for much gene expression. And then the opposite being acetylation, which is the additive of acetyl groups, allowing for chromatin to loosen up, allowing for more active gene expression. And to run these processes, our bodies use histone-modifying enzymes, which are just proteins that go in to add epigenetic marks, so chemical tags that determine the what, where, and when. And this basically determines how our genes are expressed. So it turns out that studies have actually linked epigenetic issues to things like intellectual disorders and developmental delays, which are a lot of things that we don't understand at this stage of science research and just science in general, which is why this is very important. So epigenetics is a relatively new field, meaning that not a lot of studies have gone into it, they haven't gone in depth, and we don't know much about it. Epigenetic marks can accumulate over generations, meaning that certain chemical marks can transfer to your offspring to further generations, and they can even get worse if issues do arise. And epigenetics can also influence survival, which is something that we look at in my study specifically. So before I go into the specific gene that I'm looking at, I want to go into my model organism, which is a C. elegans or Caenorhabditis elegans, and these are just microscopic worms, and the reason they're so helpful for biology research and just genetic research in general is they're simple, they're transparent, they have a short lifespan, and they actually have a lot of genetic similarity to humans. So it turns out that C. elegans have a lot of orthologs, which are just genes with similar ancestry to a lot of the genes in humans. So if we understand genetic pathways in C. elegans, it can also be applied to human health as well, which is why they're ideal for studying epigenetic regulation, which can apply to human health as well. So going into my project focus, I'm focusing on a gene called SPR2 or suppressor of phenylalanine 2, and what this gene does is it creates a protein called SPR2 as well, and this protein acts as an inhibitor of histone acetyltransferase inside of the C. elegans germline, and that's something I will go on to explain in a bit, but just in case you don't know what the germline is the lineage of cells that develop into your sperm and your oocyte, basically just the reproductive system of these worms. So going more into how SPR2 functions, so on the right we can see that inside of the C. elegans germline there are a group of proteins called histone acetyltransferases, right? And the way they work is they add acetyl groups onto these germline genes, histones, allowing for germline genes to express themselves more freely because it causes open chromatin. And what SPR2 does in this process is it takes off excessive acetyl marks to make sure that this active state turns into a repressive state, and that germline genes are only being expressed when and where they need to be. So overall, HAT and SPR2 work together to maintain proper germline gene expression and make sure that the worms are going on with their duties properly. So it turns out that when SPR2 is mutated, these C. elegans actually lose this regulatory or inhibitor function inside of their germline genes. And what does this do? Well, it means that these histone acetyltransferases are able to add excessive acetyl marks, meaning that chromatin in these germline genes are always open, and essentially, these germline genes are being overexpressed, and this has potential to cause several types of defects, one example being a chemotaxis defect, which is just a signal sensing defect. So are the worms able to sense their food? Are they able to smell it? Are they able to go towards it? And then another example is a developmental delay. So do adult worms and baby worms, are they able to properly develop? Are they able to properly grow? And then finally, offspring reduction defects. So are mothers able to actually lay embryos? Are they able to actually be fertile? And then also, are embryos that are being laid, are they able to hatch, and are they able to reach that adulthood stage? So that's kind of what my project focuses on. So why are we studying this in SPR2? Well, it turns out that there is another gene in the same family as SPR2, which is called suppressor of presenilin 5. And this entire family of genes is kind of under-researched, but this is what I found in a lot of studies. So SPR5 does the same thing. It also acts as a regulator of the C. elegans germline, except it does this by acting as a demethylase, so it takes off excessive methyl groups, and it essentially prevents hyperactivation of chromatin in C. elegans as well. And so I looked at a study that analyzed SPR5's effect on the germline. And what they wanted to see is, specifically, they wanted to look at embryonic lethality. So are embryos able to hatch, and are they able to reach that adulthood stage? And when they looked into it, this is kind of what they found. I'm going to break down this figure into two parts. So just looking at the wild types. So wild types are just worms. Wild type or N2 worms are just worms with no visible genetic mutation. So this is how the germline is supposed to function. Here we can see that this represents the 4 stages of the germline: the germ cell stage, the spermoocyte stage, the zygote stage, the 100-cell embryo stage, and then PGC just stands for primordial germ cells, which are the last germ cells in this process. So as we can see here, that normally in the zygote stage, SPR5 comes in and erases this specific epigenetic mark. So this is when SPR5 is supposed to come in and take off methyl marks. And then by the 100-cell embryo stage, these PGCs have been left with, less methyl marks, or as much methyl marks as there could be, but these methyl marks have been taken out. Whereas if we look at the bottom, we can see that even by the zygote stage, because this represents the SPR5 mutants germline, and by the zygote stage, because SPR5 is mutated, it's not able to take off these methyl marks, meaning that by the primordial germ cell stage, these black dots are still present. They're present here, they're present here, they're present here, they're present here. And, what the study found is that because these methyl marks weren't removed, this caused sterility in these SPR5 mutants. So what does this mean? Well, because SPR2 and SPR5 are in the same family, and they have very similar functions, it was also speculated that SPR2 mutants could also have a reduction in offspring. But because SPR5 showed complete sterility in the mothers, they weren't able to test if embryonic lethality was a viable reason for this reduction in offspring, which is why we wanted to see if there could be potential for germline issues and embryonic issues that cause a reduction in offspring in SPR2 mutants specifically. So this leads to my research goal, which was to investigate if increased histone acetylation caused by SPR2 mutants causes an offspring reduction via germline failure. So are the mothers able to actually lay embryos? Are they able to be fertile? Or embryonic lethality, so are embryos that are being laid, are they able to hatch, and are they able to reach that adulthood stage? And I hypothesized that hyperactivated chromatin due to SPR2 mutants caused a misexpression of genes and detectable offspring reduction resulting from both germline abnormalities as well as embryonic issues. So these are some of the materials that I used for the general gist of my main 3 tests. And this was the first test that we did, which was a brood count assay. And what this looked at was it compared N2 and SPR2 mutants, and basically the amount of offspring that they were having. I measured it specifically in embryos. And then we also took a differential interference microscopy image. And why we did this is we wanted to see if there were visual abnormalities that we could kind of pinpoint right at the bat. So we took pictures of C. elegans germlines to see if there was some difference in the N2s versus the SPR2 mutants. And then also, finally, the last test that we did was a lethality assay, which basically looked at the embryos that were being laid by these mutants. were they able to hatch and were they able to reach adulthood. So this is a summarization of our brood count data. The y-axis represents the average number of progeny, so the average number of embryos or offspring that each generation was having. And yellow represents the N2 wild-type worms, whereas black represents the SPR2 mutant worms. And one thing to note is that before generation 9, this is kind of when the C. elegans are still recovering from getting out of their frozen capsule state, so that's kind of why the data is a little wonky, but not too much of an issue. So as we can see here, if we just observe the N2s, on average they have around 300 to 400 amount of offspring throughout all generations. Whereas if we look at SPR2 right off the bat, we can see that the average number of progeny stays around 200 and below. So here we can kind of see that in these SPR2 mutants, they aren't acting normal. They aren't having the right amount of offspring that they should be compared to the wild types. And then if we look at the overall trend, we can see that while it is a little bit wonky, overall these N2s have a horizontal trend, so they're relatively having the same amount of offspring throughout all generations. Whereas if we look at SPR2, there's kind of a clear small decline in the amount of offspring that these worms are able to have, and that kind of shows that as generations go on, these epigenetic marks that are caused by these SPR2 mutants are getting worse, and the further generations are not able to have as much offspring as the previous. And so this is just the germline photos that we took. So the top two represent the N2 wild types. So here we can see a picture of the C. elegans germline, and these are embryos inside of the germline. And as you can see, they're all relatively the same size. They stay in a uniform line kind of structure, and this is how the germline is supposed to be at this stage of life. So specifically the L4 worm stage, which is just how old are they? And then here, this is a picture of the spermatid region of the C. elegans, which is just where sperm residue is supposed to be stored at this stage of the worm development. Now, if we look at the SPR2 mutants, we can see that inside of the gonad, there is no embryos. In fact, these are embryonic cells. Now, both of these worms are at the L4, growth stage. So at this stage, these cells are supposed to be embryos. And we can even see here that they aren't even in a uniform line. They're kind of, jumbled up. They vary in size. And that's how you can kind of see there's already a visual abnormality. And then in addition to that, it's kind of hard to see, but there is white residue, which was actually found to be sperm residue. And like I said before, this residue is supposed to be stored in the spermatid region at this stage of growth. So this kind of shows that there were already visual abnormalities in the mothers of SPR2 mutants, showing that, germline issues was one of the reasons for that reproductive, offspring decline inside of these mutants. So this is our lethality assay data, which I've kind of summarized into one graph. So the y-axis represents the percent of embryonic survival to adulthood, and the x-axis represents, N2 versus SPR2. So as you can see here, about 48% of the SPR2 mutant worms did not actually hatch or reach that adulthood stage, whereas if we look at the N2 wild types, almost 100% of them did. So what does this mean? This means that embryonic lethality is present, and it's part of the reason that there is an offspring reduction. So it's not only germline issues in the mother, but it can also be embryonic issues. They aren't able to hatch and they aren't able to develop. So for overall data findings, I found that a reduction in brood count is due to both germline issues inside the mother and embryonic issues in the embryo or egg itself. And this is really important because lethality or embryonic lethality has never actually been proven in SPR2 or any other gene in the same family. And then also, this is, one of the few studies that connect hyperacetylation or just acetylation in general to issues like this in any type of organism. So for some implications, this is the first study to connect epigenetics to lifespan. Because epigenetics is such an under-researched field, not a lot of people have found correlations like this, and this kind of gives us an insight into the scope and role of epigenetics, because not only does it cause intellectual disorders and developmental delays, but it goes beyond that. For those who don't have that, it can cause issues to lifespan. And then also, these can also be applied to human health. So, like I was talking about, C. elegans have a lot of orthologs to humans, and specifically, there are a set of genes inside of humans called SET. And the way they work is they also work as NHATs. And it turns out that when they are mutated, they've caused a lot of intellectual disorders and developmental delays in a lot of those patients. And that's why we think that studying SPR2 and this family in general could give us a framework or could contribute to the knowledge within therapeutic strategies for issues like these. And that's kind of why it's so important to understand just frameworks like this. Thank you.